so cool. We can actually see things in the human body. Even the 7 Tesla, there might be 1 or 2 clinical applications only.
Okay.
Today's
interesting. Probably in research, this heterogeneity of the findings and the methods in which they've been applied, it complicates everything essentially.
You're the boss. The first experiments are going to be done on your head. Doesn't mean that this is going to go anywhere near the clinic anytime soon, though.
We have to be honest about this. And I think radiographers on research side normally get paid way less than the clinical ones.
It's a mess. If I'm on a G, I'm using that method. On a Philips, I'm using that. On a Siemens, I'm using that.
Me as a radiographer, MRI radiographer for 12, 13 years. At this point today, I wouldn't know how to get into research if I didn't know someone that does it.
It's one of the few medical imaging techniques that passes the test of your grandmother and your daughter.
The research radiographer as a pathway, I think, requires a little bit further implementation.
Look, guys, you're dealing with a very low signal to noise method. What we need is signal.
It doesn't happen quite often to have the opportunity to sit on a table with someone that developed such a passion for imaging and research to be awarded with titles like Honorary Professor, Director of Imaging Services. So I think it's nice to be here with you, Xavier, today. And first thing I would like to know, what were the specific reasons that bring you to develop this interest and also with the years this knowledge about research and specifically imaging and neuroimaging in this sense?
Well, thanks, guys, for having me here. I guess the original interest I got to work in MRI came by chance encounter during my studies of physics in Lausanne in the early 90s. A professor from Zurich came and gave a talk about MRI. And I was interested in going into medical physics, and these early images that I could see and even videos created, and that was actually played on a video cassette of a heart measured with just MRI moving like this. It just actually blew my mind, and I thought, wow, this is so cool. We can actually see things in the human body. And right after my studies, I actually applied for a PhD fellowship, got it, and went to do my PhD with this group. So, and from then on, it's been love at first sight, as they say, and I never turned my back to it. So, yeah, that's how it came about. I had a relatively interesting career moving in and out of academia. I've worked for major MRI manufacturers as well as actually working for research centers and universities. So it's an interesting path, not the traditional linear path.
So you've always been involved in research throughout your career?
Pretty much, yes. Pretty much. When I was working at some point I was working for Philips, and I was actually employed as a clinical scientist on a site in Singapore for 2 years, where I was working there for Philips. And over there I was developing new methods for measurement of perfusion in the brain.
Oh, there you go.
Stuff like that. So it was always a bit about research-based stuff.
Can we give to the people at almost an understanding of what have been the key themes and areas of your research and your contribution as well to the MR field?
Yes. I'm not sure I had that many
contributions. Don't be humble. Don't be humble.
No. Well, I started my PhD in 1994. Now, in 91 came this brand new technology that for the first time ever, we could use MRI to measure a surrogate marker of brain activity. It's called functional MRI. Yeah. And that came out with 3 papers from 3 different groups in the US in 1991. by 1994, in Switzerland at least, there was actually nobody really using that method. So I started my PhD trying to develop the whole setup in Zurich to use and to be able to use fMRI, as it was then called, to try to understand what we were measuring, etc. So in the context of trying to understand what we were measuring, what was this signal that was actually representing a brain activity of where you were moving your right hand, you would see some signal on the left motor area of the brain, etc. I developed an interest for the measurement of blood flow and the perfusion, the delivery of nutrients to the brain, which increases upon activation. And this is basically what you're measuring. And I guess the first part of my career, for the first 10, 15 years of my career, I've been working on methods to develop perfusion methods. So basically methods to image the blood reaching the tissue. Now, after my PhD, I got sidetracked away from pure academic neuroscientific research into more applied clinical research. So using these methods in diseases like stroke or dementia or this kind of method. In the context of trying to understand what was going on in the brain, I got interested in neurophysiology, so measuring exactly what's happening in terms of the metabolism. So the blood flow is one, the oxygen usage is another one, but at the base of it all is about sugar and how actually glucose can be used by the cells to actually generate energy, and that was one of the biggest interests. So I guess the second part of my career was I got interested in an advanced method. I'm not sure whether you guys have heard about. It's called chemical exchange saturation transfer, or CEST.
Yeah.
And
I actually wanted to ask you what it is and how it works. So segue.
Okay, so CEST is a method in which you're trying to use NMR tricks to increase the signal in situ of a very tiny amount of stuff. So basically, if you have glucose, glucose molecules has, for example, some hydroxyl atoms. So these are hydroxyl bond is OH groups, and these OH groups, these protons are in exchange with the surrounding water. So if you keep saturating these protons, they are slightly off resonance with respect to the resonance of the water peak. If you keep saturating those, you eventually, through the exchange process, start to diminish the signal in the of the water, which is basically the main signal that you get from MRI. And by doing this, what's happening is you can actually indirectly detect a small amount, a few millimolars of a stuff. It could be different types
you target different molecules. You target
you could target different molecules. You could target different chemicals, so and different bonds and etc. Now, the problem with this method is like you increase a lot of sensitivity, but you lose a little bit of the specificity, because actually, glucose is not the only molecule that has hydroxyl groups. There's loads of molecules that would have a hydroxyl group. So it's a bit complicated. It's still a research a research thing. So, but in this case, we could actually perform an experiment in which we would scan. In this case, we started with rats, mice, so animals, where you could scan the brain of the animals before and after injection of glucose, and you could see an increase in the signal.
That's interesting.
So that you could actually just use as very standard glucose, like 𝐿𝐿𝐶, really, drinking, and basically trying to see an increase in the signal directly in the brain. But the brain was not the most interesting organ, it turned out, because one place that uses a massive amount of sugar are the tumors. Basically, any neoplastic tissue, because of the biochemistry of it, will actually start to use way more sugar than the surrounding tissue. And that has been used for a long time using PET, FDG PET.
Sugar has been connected to cancer for many, many years.
Exactly, exactly. Actually, since before Second World War. A guy called the Warburg effect. It was the first one who actually discovered the fact that a cancer cell actually feeds off much more glucose than a regular cell. So with this was basically another big tranche of my career has been about trying to develop this method and to bring it to the clinic, although it has I've been faced with a lot of troubles, and as many other groups that have tried. And I was very, very pleased. I was invited last week to the 25th anniversary of a research center at Johns Hopkins and Kennedy Krieger Institute in Baltimore, where I started my career as a young postdoc in 1999, so 25 years ago. And where I saw the first really good results, reproducible results in patients with brain cancer of this method. But, it took a lot of time, like around 15 years until we got the first results. It doesn't mean that this is going to go anywhere near the clinic anytime soon, though. And that might be some of the things we might discuss about.
I think this is a perfect access to what we would like to ask you, because I think what we wanted to know from this podcast episode is like, what are the challenges? I think you are the right person to answer this question. In this transition, the challenges in this transition between research and clinical practice. You mentioned research on small animals, but how do you translate that and those results to say, yes, we can safely implement this practice into, humans and whatever?
Well, the answer is very carefully. But in addition to that, no. I mean, so if you're injecting something into an animal or a human, like in this case, even glucose, which is very safe, any kind of translation will require a very, very rigorous ethical approval. You need to file this as a proper clinical trial and etc. And it takes a long time. Yeah. It's a very arduous process. But beyond that, a lot of the methods, like the method to measure perfusion, were not really developed widely on animals before they were translated.
Is that the ASL?
Yeah, it's called arterial spin labeling, because the method is not following an injection of a contrast agent, which exists as well, but it is actually, you are inverting the magnetization that is in the arteries. You wait for it to get into the organ of interest, in this case the brain, and again, a phenomenon of exchange. This arterial blood, which has this inverted magnetization, will exchange with the local tissue water and will reduce that signal. And by measuring it with and without labeling, you can actually detect the perfusion, the amount of blood that has got out of the arteries and into the tissue.
Okay.
So this is the way it works. And this because you don't inject anything, the translation in principle, translation from I'm trying that on a bottle to I'm trying that on a healthy volunteers, usually myself or one of my students.
So it's always you. You play the key role in that transition.
Absolutely. Yeah. Like, you're the boss. The first experiments are going to be done on your head. I have
traditionally like that.
That's traditionally.
Do you have to sign a waiver in case anything goes
wrong? No,
I
mean the case. It's more interesting than that. I cannot ask my student to do it, right? Because that would be seen as coercion too. Because there is there's a relationship of authority here. So we need to be very careful with that. So you have to find somebody. So usually, once you think that it's safe enough, Generally, it is. You go and put your own head in
I
there.
like your optimism.
I think we need a little bit of this in research.
Yeah,
Otherwise,
yeah.
we won't obtain any result. We'll
Yeah,
say,
yeah.
it will work. It will definitely
It
work.
should work. Anyway, and then you go in and well, I mean, it's not that easy. It requires ethical approval, etc., etc. So don't take me wrong. It used to be easier, I can tell you. In the mid-90s, it was not that not that strict, but it got more and more strict, which is a good thing because it actually helps avoid problems in general. Anyway, this translation is relatively easy as long as you don't as you're manipulating spins, as it were. So like you're just using the MRI in a slightly different way that it's used usually, but without much difference. That is an easy translation. The biggest problem is from having published a couple or more papers in a few patients to start to get this to be used clinically,
Yeah.
let alone in
That
research
jump
center.
to the
that
clinical
jump.
So yeah, that's the gap from preclinical to clinical is relatively easy. If you're using a small animal, and for CEST, it was more complicated because CEST works much better at very high field, which is easy to have on a small animal.
When you say high field, we're talking 3, 7, what are we talking?
Well, actually, the original experiment were done on a 9.4 Tesla small animal scanner.
9.4.
Okay.
Yeah.
Wow.
I was in the middle. No.
Yeah,
I was gonna say 11,
these
but-
days there are about 127 Tesla in the world.
Yeah.
And a few, like a handful of higher field strength between 9.4 and
There's a 14
10.5.
coming as
Yeah.
well.
There's a 14 coming up
Only-
in the
obviously-
Netherlands. There's
Yeah.
there's a
11.7?
12 Tesla that's going to go up
Just to be
in
very-
Nottingham.
Just to be very transparent, these are only for research purposes at the
Yes,
moment,
yes.
right? Only.
Yes.
Yeah.
Even the 7 Tesla, there might be 1 or 2 clinical applications only
Okay.
today's day.
Interesting.
Which can be used and have an actual reason of why you would like to pay for that amount, because a 7 Teslas are 6-7 times more expensive than a 3T. It's
Wow.
really expensive.
And those applications are neuro applications.
Yeah, they're only neuro.
Yeah.
Also, the current 7 Tesla scanners are generally don't have a body coil. So you have a send received or transmit receive coil for the head or for any the knee or any other organ, but you cannot actually do cardiac MRI very easily. I think there's 1 or 2 groups that have developed body coil for 7 Tesla, but it's a complicated.
Yeah.
It's extremely complicated, and I know you guys talk a lot about MR safety. At 7 Tesla, the RF deposition is very high,
Astronomical.
so
Of course.
it goes square. The amount of power that you deposit into a body goes square with the field.
Yeah.
So
Yeah.
at 7 Tesla, you need to be very careful.
To go back one moment to the transition between the research to clinical, is that anything you would suggest in terms of protocol validation? Something is like a structure that you say to get earlier these results, or more accurately or more effectively?
I don't know. As we were saying, the transition from preclinical to clinical is okay. From early experiment in the clinics to being widely used, takes a next level amount of work.
Yeah.
If I take the example of ASL, it's a very good example, because ASL, in 1991, you had fMRI. ASL was developed in 92, right? So next year. In the 90s, I was one of the few people who were working on developing different methods, new methods, improved method or not, I don't know. But, trying to get the techniques moving. By the mid-noughties, it was working quite well. But because you had, I don't know, a a dozen or more different academic sites that have developed their own method.
Mm.
Each of them working more or less with a manufacturer. Each of the manufacturer by the mid-noughties having, taken on board one or the other of the methods that they were developed by one of the people, one of their luminary sites, and having implemented that, you ended up in a position where most of the people who would like to start using it saying, it's a mess. If I'm on a G, I'm using that method. On a Philips, I'm using that. On a Siemens, I'm using that. I'm reading all these papers and they all say something else. And there's no harmonization into any of the acquisition protocol, the readouts. Nothing is actually really clear, even because this kind of measurement, if you want to get a quantification, a quantitative value, you need to actually have some input into a certain model. So you had maybe a dozen methods of labeling, at least 3 or 4 different types of readouts, and just as many, like around a dozen different models.
This reminds me AI as well, the modules of AI, the algorithms are all different. And
there's all sorts of
plenty.
Except that AI in general, you don't know what's going on. Here, all of these were very traceable, if you want. You knew what was
exactly what they're doing. Yeah.
So right around that time, together with a colleague from Germany, Matthias Günther, we put together a group which we called the ASL network. It's like, okay, arterial spin labeling, we are going to move this into the clinic. And that was our goal. Like, as in really being used clinically, not demonstration. And so we got together, we managed to get some funding from the European Union, a so-called cost action, which is basically doesn't give you any money for doing the research, but gives you a lot of money for bringing people together, eating and drinking together. Great things. Yeah, it's the first very large grant I got was actually money for aperitivi and a nice meal in great places.
Did it work at least?
It worked fantastically. Look, the result of those was actually at the end of this big 4 years brainstorm and working together and having young students spending 2-3 months into another group. So you start learning the methods and appreciating what is best in which group, and at the end you can bring all of this together, and together with the International Society, the ISMRM's Perfusion Study Group, we came up with a white paper which says, okay, now guys, if you want to use ESL, we recommend you use this protocol with this model, with these values input into the model. And that paper that came up around 2010, I think, if I remember well, really changed the field, because with that, it was a grassroots kind of approach.
They set ground rules, basically.
It set the ground rules, but from the specialists, including neuroradiologists, physicists, and I think there were a few radiographers, but too few, unfortunately, and we could discuss that later.
That's a question.
Yeah, exactly.
But these were there. So following that publication, it was really humbling, because we were quite a few people, each with their own method. I had my own, and other ones, but we really listened to the neuroradiologists in a group who were saying, look, guys, you're dealing with a very low signal to noise method. What we need is signal. So it came very quickly for us to, the decision to actually go with one method over another became very easy, because we just had to choose one that had the maximum signal. And it wasn't mine. But everyone, it wasn't. No, that's research, right? So
Yes,
it was some
exactly.
other. There were also some other people, and everyone. These people, when a researcher has developed something, it's almost like their baby. They are really, really attached to it. But I thought this community was amazing because we managed to get them together to make a decision, and for one winner of one method, there were maybe again almost a dozen losers, right? Because this is their recommended method. All the other ones were there
But
for.
at the end of the day, those other ones helped the one that won.
Exactly.
Yeah,
to
exactly,
be the one
exactly.
chosen,
But
right?
but people could get together, and with that, all the manufacturers within the next 5 years or something eventually changed their implementation to follow our guidelines.
Perfect.
That's awesome.
So that was the very first step.
So
It's not necessary. It's not enough, but it's a necessary step. You need to be able to have the same method that is
to apply that.
being able to use and buy the key, buy the software, whatever it is, from any one of the manufacturers and say, I'm going to use these in order for it to be able to start
This is a recurrent
being used.
issue on MR in general. Like MRI safety is the same problem. It's not standardized. The names of the sequences are all different because each vendor has their own kind of thing. So for us as radiographers, it's always a struggle as well because we're working with Siemens and tomorrow we're working with 𝐿𝐲 and the next day we have an Hitachi scanner. And it's very confusing for us as well. It's limiting the way we can develop our careers as well.
Absolutely.
Maybe for us it's more like terminology problem. But I do agree that probably in research, this heterogeneity of the findings and the methods in which they've been applied, it complicates everything essentially.
Absolutely.
We don't find, that middle ground. It's difficult to be.
It's a very interesting thing. So in this case, we managed to get that going. But it doesn't mean that ASL is used in every neuroradiology clinic today or anything. Because once you have that, then you need to see, okay, in which particular disease will this method actually make a difference? A difference in the diagnosis, a difference in actually the patient management. Because if you don't have this, it's likely that the neuroradiologist said, okay, look, I have just that amount of time to scan this patient. I'm only going to include additional methods. So,
you have your toolbox. And
you have your toolbox. You can choose. But in Europe, it will be done based on best practices. In the US, for example, you will need to have a reimbursement code.
Yes.
And without a reimbursement code, even if you have the best method in the world, it's not going to be used. So there is a hell of a lot of work to do from developing your most amazing new MRI method all the way until it gets into the clinic. You need to make sure that you have a similar method across vendors. You need then to make sure that there is the application are warranted, and that requires clinical trials or at least large numbers of patients being scanned with one method or another until it can actually, in individual hospitals, starts to be used.
And in US, sorry to interrupt you, with the clinical trials, it's also harder to do it because they want American patients for those clinical trials.
True. That is if you want to get a method that is FDA approved.
Yeah,
absolutely.
So you can't just have something clinical trial, let's say in Portugal, and then bring it to FDA and get it approved. You need to do it in an American hospital with American patients.
True. That's an interesting point. However, that is if you want to get a method, so-called de novo method in the US. So basically, if you want the FDA to approve a method that's never been done before.
Yeah.
For a lot of things that have been done with MRI, you can always try to convince the FDA that something similar has been done before. And if you can convince them to do that, then you can go another route. So it's like not a de novo application, it's typical American.
Example,
there is called a 510K route. This is a typical American, numbers stuff. But what it means is, that this is a procedure that you follow that actually allows you to tell, We're barely doing what has been done before. This is just a new application. Similar. Please give us the...
So for example, if you have CE mark, it will make it easy for you to actually get the FDA
yes,
approval of your
no,
module.
Actually, these days, if you have a new medical device, because of the MDR, the medical device regulation, the new regulatory pathway for medical devices in Europe, it's so complicated.
Yeah, it's easy the other way around.
So these days, while it used to be with the MDD, the Medical Device Directives, before this new regulatory framework, it was easier to get a CE marking and then to go to the FDA. Now it's the other way around. In principle, you start with the FDA because it's easier, and then you go to the MDR and to get a CE marking.
So different pathways on which tends a little bit to complicate things, but we're working on that based on my understanding.
Yeah,
anyway.
we are. Yeah.
Behind the scenes, before you mentioned, you told me something that, to be honest, I was thinking about. Why did he say this thing? Because coming to you had a certain impact on myself. Don't worry, nothing too serious. You told me that you think we need more radiographer in research in general. And is there a specific reason why you told me that? Because I don't hear this quite often, and as you mentioned, sometimes we tend to be, just forgot from the multidisciplinary team.
Yes.
So would you like to tell me why you think we need more radiographers, more MR technologies, more MR users in general?
Generally, there's a bit. Let's put it that way. If you are developing a new method, and that this new method is extremely complicated to use, it will require a lot of very complex settings and etc. You might not actually get the buy-in from the radiographers. And you guys, I'm sorry to say, but the buck stops with you. You have the best method in the world. It's too complicated, or it's something that is actually, when you are in research and you have a PhD student who is working on that one thing for 4 years, they become the world specialist in that domain. And for them, it's quite normal to think about these 120 different things that they need to do to get this method to work. But if nobody takes that to actually bring it to a point where it can be done in a really simple way, there's no way that it's going to be used in the clinic. Just like with everything else we talked about, on the one side, the regulatory framework, FDA, CE marking stuff, there's the reimbursement, best medical practice in the UK, NICE guidelines, etc. This is another aspect about getting something new. And that requires harmonization and making sure that the quality of the scans, the quality of the MRI scanners, etc., are all set properly. Yep. But then there's the other aspect, which is the simplicity. And a radiographer is work is actually a very hard work. And you guys have a lot to do, a lot to learn in an ever-changing field. And if you're not actually involved in the whole process, it's sometimes very hard for an academic or somebody coming from research to think about how can I make my methods become easy enough so that it's not going to give an additional headache to the radiographer.
Fantastic. This is very interesting.
And that's why I was saying to you, that's where, including radiographers and MR technologists early in the translational pathway can be very, very important. In addition, there's the programming brand new pulse sequence and stuff and etc. with an MRI scanner. That's one aspect of the research. There's a big other aspect, which is about how can we optimize protocols and stuff and etc. And this is really also very important because you can gain a huge amount of signal to noise in your very special complex methods or something if you do that properly. And again, I've had many radiographers as PhD student who went on, and their work was partly to optimize protocols and partly to actually do the translational work, which is let's go and start scanning 20 patients with brain tumors using this ASL sequence, this 𝐶EST sequence or something like that as part of it, analyze the results and tell you, is it something that it can go further? So I really believe that these interactions with radiographers is quite important.
It's essential, yeah.
It's
Do
really,
you
really...
think there's a lack of interest from the radiographer side to join the research? For example, from my group of friends, they're all radiographers, MRI radiographers. They never mention, I would like to do research. They're like, I would like to do cardiac, or I would like to do breast MRI or biopsy, something like that. Actually, I think you're the only one that I know that actually loves research. So you can speak about
actually,
this better.
I would like to respond to your question, and I'm very interested to know your point of view as well. I think is rather than there is the potential lack of interest as well, but it's more lacking of opportunities to me. You mentioned radiographers doing PhD, and I want to challenge those at home. How many of you are aware that there are PhD opportunities for radiographers? I think very little, to be honest. And because maybe there are very little opportunities indeed, but maybe I have the interest, to know more about research, but maybe no one kind of like properly expose me to this field and telling me that actually there is a pathway for you to develop your own research and become
road,
exactly.
It's a two-way road. Yeah, absolutely. I think for radiographers to be potentially also getting more in contact with the medical physicists, with the researchers, this can mostly be done via the learned societies. For example, the International Society for Magnetic Resonance in Medicine, the ISMRM, has a particular direct relationship, or I'm not sure how it goes, but it's jointly with the equivalent for technologies, the ISRT, which technologies is just the American term for radiographer. And the ISMRM is always have a joint meeting with the ISRT, so they're all together. And during these meetings, while they have their separate sessions, everyone can attend any sessions, and it's quite an important forum. But then there is another issue, and that is something that's probably you would know that because you're both not of English mother tongue, but a lot of radiographers, because of the way the whole field works, are usually speaking their own language with their patients and something. And quite a few might not be that fluent in English. They don't need to be for their job. And that is sometimes of a hurdle because the whole research, all the publication is in English. The places where you have a lot of interactions between radiographers and the international community
language
is all in English. And I find this, it took me a while to understand how much of a problem that can be, because it's a very real world problem, right? Most of the people who are in the leadership of this the ISRT are coming from Australia, the US, or the UK, because that's their native language. You really rarely have people from one of the radiographers societies that rise to this kind of post unless they are very, very good in English. And it is a problem, and that means that a lot of these efforts will need to be done on a country-by-country basis.
Yeah, I think that's a big issue. There's something that I'm trying to start in Portugal as well, trying to get those people together and build something within my own country so then we can scale up. But I also think it's a problem of access. For example, me as a radiographer, MRI radiographer for 12, 13 years, at this point today, I wouldn't know how to get into research if I didn't know someone that does it. So for me, it feels like, and for my peers as well that I speak about this, is we always feel like we can only get into that field if we know someone that does that field. So for example, if I wanted to do research, I would ask Julian, how can I get into it?
Yeah.
Because Julian is already within that kind of field.
True.
Otherwise, I wouldn't know how to do it. I wouldn't know who to approach, where to go. I think there's a lack of information outside for the radiographer class to guide them a little bit. I don't know if you went through this, but to be guided like, this is the opportunity that we have.
And I think this starts from the bachelor, to be honest. So no one tells you exactly that there is this opportunity. Even when you do the master, then in that case, you start to get a little bit more exposed into research. But if we don't start from the basics, if we don't start from the fundamentals, meaning from the moment you start, you can also be a research radiographer as a pathway, then this will be automatically excluded for some, meaning I'm not aware. I can do X-ray, I can do CT, I can work in MRI. My pathway stops here. So the research radiographer as a pathway, I think, requires a little bit further implementation.
Most likely, yes. Yeah.
This is something that actually, we're releasing a cardiac course soon, and one of the lectures that we have there is exactly about this, and Julian can attest better about what I'm talking about. But on the advanced module, we had 3 modules, but on the advanced module, we have lectures just about how to become a cardiac researcher. And it's just to bridge this gap as well.
Yeah.
And I think it's important to do these kind of conversations and this kind of teaching, because we as radiographers normally don't find these pathways, as you were saying.
Imagine in field like cardiac MRI, this is crucial. I mean, performing those protocols, you mentioned before how CMR plays a key role in research, but knowing,
CMR is one of these methods where almost all of the quantitative MRI methods that have been developed have been translated to the clinic. Well, unfortunately, in neuro, it's not the case. Right?
Yes, that's one of the points I wanted to touch.
We could discuss that. Just one thing about the research radiographers. Most people I know who have started a PhD in the field happen to be working at some point or another for a research site.
Yeah, exactly.
So you're working with the researchers and etc. And at some point, you can actually just ask around you, one of the professors, one of the lecturers, if there would be a project for you to work on. And this works quite well. In the UK, you'll probably be able to have relatively low tuition fees because it's a graduate school, so universities ask for money, but this can be actually often, you can find a solution with your employers about that if you're working in a research center. So if you were interested in research, if this is something that is of interest to you, I think one of the first step would be to try to find an academic center, an academic hospital, and see whether you can join and start getting involved.
And a supervisor as well, I think.
And then, once you are in a place, it's probably easier to find a supervisor. You start doing a whole lot of different things, and one might pick your interest and you can go on with it.
One of the things, sorry, Julian, that I also think it's hindering radiographers to go that way, I think it's the salary. We have to be honest about this. And I think radiographers on research side normally get paid way less than the clinical ones. And I think that's also an obstacle for someone that is starting their career and they're like, okay, I'm 22, 23, I want to make some money so I can enjoy my life, let's put it that way. Or you want to build a family or something like that. And if you go to research, you get paid way less. And I think that's also not optimal.
I just want to add something on this because it's perfect. And it's not only that, it's definitely this, plus the fact that if you really want to go into research, what about funding? Because funding opportunities is so limited. And I got told that I can also accept to pay myself for doing the PhD study and then looking for funding. But for start, paying myself, I was like, no, I mean, in my view, I'm kind of like giving all myself into research, so I should get something back. It's not me that I should pay to do research.
I know, it's terrible. I mean, don't take me wrong. The whole research field and the way it works, I mean, it's for another podcast, but it's the continuous struggle for funding. It's real. And a lot of the radiographers I had as PhD students, for example, many of them were coming from Saudi Arabia or from the Middle East with stipend from their government, because the government there wants to invest in them.
Absolutely. It's a cycle. I think
and then they get their PhD, they go back, they get a better position, and etc. So it's an investment. And as long as the governments here don't understand that also for radiographers, it's not going to happen, right?
Absolutely. I think we're almost done, but I think we want to touch one more point before we close. We have one last question for you, but this one comes before that. I just want you to tell me a little bit about the gold standard phantoms. I think it's a company that you founded, and it's really important in terms of how we reproduce the same results within the MRI machine. So if you have 2-3 minutes to let us know how it works.
Absolutely. So this company is 10 years old next month. So we started it on the back of this big grant I was talking to you about, to bring people together about arterial spin labeling. And our first product that we made was actually an ASL perfusion phantom. And originally we thought we were going to revolutionize the field with that, and etc. Well, it turns out it's a very complex phantoms, very expensive, so it's difficult to sell as well. But never mind. From then on, we actually set up an entire range of quality assurance devices, because it seems that, whenever you were trying to measure something with MRI, it's actually a good idea to have a reference values that you can actually, have in every site, and for which you have a ground truth. So if you're going to measure T1 using your Schmolly-Molly method on cardiac, and you want to know that you're measuring the same thing at King's College Hospital as you are, at Hull teaching hospitals, you might want to actually scan these phantoms in different places and make sure that you measure the same thing.
Absolutely.
That's, so we've established ourselves as one of the 2-3 leading companies in the world providing quality assurance devices for quantitative MRI, as well as, my very first love, which is fMRI. We have now this method that enables, this product that enables sites to check both the RF chain and the gradient stability, so that you can reduce the scanner downtime and making sure that you get the preventive work done by the companies in time. So all of this is actually right there. And very interestingly, the reason why I'm here also in London this weekend and then on Monday is trying a brand new device that we've developed, which I'm very proud of, which enables to do calibration of the MRI scanner. So if you're using a scanner to measure, in this case, ADC, diffusion in prostate cancer on one system, you can get the same reproducible measurement on every system. It automatically corrects.
That's a predict project.
That's a predict project. It automatically calibrate the results, so corrects for bias and estimate actually the uncertainty on the measurements directly in every pixel. So
this can have a massive impact
I
on the
think
MRI field.
This will have
speaking
big
about MRI, what about
impact.
our final question?
Yeah, so we have a question that we ask all our guests, and basically is why MRI for you? I think you kind of answer it at the beginning, but where is your passion from MRI comes from?
It's an amazing technology. As in, it sums up a number of amazing technologies right from the start of having a superconducting, extremely powerful magnet all the way to having, methods that can actually be further and further improved to test not only the anatomy, but also the function of organs all the way down to the physiology. I mean, I don't think there is any other method out there in any other imaging method that has such a wide range of applications. So it's just an ever-evolving playing field. So and for this, it's great.
And I can see you're a curious man. So this is the perfect thing.
Exactly. And then also the next thing is, it's one of the few medical imaging techniques that passes the test of your grandmother and your daughter. You can put you can feel that it is safe for you to put your grandmother or your daughter in there, like your young
I never
child.
thought it from this point of view. This is really interesting.
So it's something that it is safe. You guys talk a lot about MRI safety, and for good reason.
I normally say it's not safe, so I like to see.
No, but if you're looking at it from a slightly different perspective, CT, X-ray, PET,
it's a very good
all of
argument.
these have ionizing radiation. You don't want to have somebody having repeated scan on there.
Absolutely.
Yeah.
But MRI, there's no biological effect. As long as you're using it within the safety limits of how it's supposed to be used, it is extremely safe. You can do it again and again and again. And I think for that reason, it makes it also more fun.
That's why we're all here.
Thank you so much, Xavier. Take care. I think that this was the perfect ending of the podcast episode.
Thank you, guys.
Just ask you to leave us a comment.
If
that's okay with you. Just anything you want to tell Everything MRI about today's experience.
Absolutely.
Take all the time you need, and thank you
Thanks
again.
for the invite, guys.
Thank you so much.